So thank you again for that introduction. A little bit more background about me. So I did my PhD and my postdoc in the Dahlman Lab at Georgia Tech, which is where I learned about drug delivery and kind of built a lot of platforms that allow us to screen different lipid nanoparticles in vivo. And so what I'm going to be talking about today is a new technology that we recently published on at Nature Nanotech, and is the basis of the technology that we're licensing into Nava. So this technology is known as SENT-Seq, or single-cell nanoparticle targeting seq. And the reason that it's important is because it allows us to address a lot of roadblocks that are traditionally found in delivery. And so, I'm going to talk about some issues to hepatic delivery that a lot of people are probably, or to non-hepatic delivery that a lot of people are probably aware of. But these issues aren't just for non-hepatic delivery, they're just general issues in delivery that I think are roadblocks for a lot of folks trying to find good lipid nanoparticles. So the first of these is that systemic clinically relevant RNA delivery is largely limited to the liver. And so what that means is that we don't have a good case study for non-liver delivery. And so there isn't really an outline that, companies or academics can follow that allows us to kind of easily bring a novel lipid nanoparticle that goes to a non-hepatic tissue, either, to a non-human primate or eventually even to patients. And so there's a lot of folks that are, doing good work in this area. But I think, there's still a lot more that can be done, and we're still kind of at the very beginning of non-hepatic delivery. Part of the reason why it's so hard to go outside of the liver is that a lot of the screening methods that are currently out there often rely heavily on in vitro testing, which our lab found, at least in the case of biodistribution, does not correlate with in vivo delivery. And so if you're trying to screen for a lipid nanoparticle that delivers to lung endothelial cells, the most efficient way to be doing that is to be looking for delivery directly in lung endothelial cells in the animal model that is most predictive. And so there's a lot of even questions there. What is the most predictive animal model for humans? And, a lot of us work in mice. Is that the best model? Is it non-human primates? That's something that I think, time will basically show. And finally, so another kind of roadblock is that rare cell types can often be difficult to characterize using traditional methods. So think of flow cytometry or any other like cell sorting based method. And the reason for this is that you might be able to stain for those rare cell types, and maybe you can flow on them and record enough information to figure out if you have delivery. And by that, I mean functional mRNA delivery of your payload. But in order to actually screen in these cells and figure out what's going on in these cells, a lot of times you have to be able to isolate them. And so doing that with fluorescence activated cell sorting or other cell sorting methods can often be incredibly inefficient. You might have to enrich using other markers. So it gets really complicated really fast. And then finally, a lot of the current screening methods for lipid nanoparticles often rely heavily on reporter mouse models, which prevents screening in disease and humanized models. And for this last point, there's been a lot of good work that's been done to kind of get away from these reporter mouse models and move into more species agnostic mouse models. And I'll talk about that a little bit down the line as well. So given that there's all of these roadblocks, how do we actually discover viable and efficient LNPs? So I like to show this slide for two reasons. One, I think it's always good to kind of conceptualize what we're talking about in terms of lipid nanoparticles. And two, I also think it's kind of fascinating that LNPs can be made from 2 to 4 components, and you can interchange these components in an almost limitless way, depending on, how many different synthetic compounds you can make. And so this really opens the door and kind of allows you to screen unhindered in a way that's kind of just open to imagination. And I think that that can also be really, really stressful as well, because you have a lot of different options and there's no good way to kind of understand which options for phospholipids or ionizable lipids or PEG lipids lead to delivery to a particular cell type, because we just don't have enough lipid nanoparticle candidates out there to kind of draw those kinds of conclusions. And so, I think while LNPs have a lot of potential, unlocking that potential is something that's definitely difficult. So the way that we kind of approach this problem is we developed a few different screening systems in our lab back when I was a first and second year PhD student. And one of them is known as FIND, or fast identification of nanoparticle delivery. And what FIND allows you to do is that it allows you to pick for LNPs that functionally deliver their mRNA payload to the cytoplasm of a cell. So that means you're picking for particle 3. Shown on the right side here. And you are avoiding or kind of not selecting for particles that are either stuck inside of an endosome or stuck on the outside of a cell, like particles 1 and 2. And so what that allows you to do is really hone in on which particles functionally deliver their payloads and ignore all the noise that comes in the background. This was a big kind of step up from our biodistribution screens, which wouldn't be able to tell the difference between the delivery of these 3 particles. So the way that this system works is that particles are formulated so that they carry an individual single-stranded DNA barcode and a particular mRNA. In this case, we were using mRNA, which is reporter mRNA. And each particle was formulated individually. And then particles underwent quality control criteria and were pooled together. In this pool, we included something called a naked barcode. And so this naked barcode is unencapsulated barcode, so unencapsulated DNA, which shouldn't readily cross the cell membrane. And so this naked barcode kind of acts as a negative control. And we can look at that in our data analysis downstream to know whether or not our experiment is successful, or at least whether or not our experiment kind of has any problems with it. And so this pool, including our negative control, is injected into AI-14 mice. So these mice have a lock-stop-locks TD tomato cassette, such that when a lipid nanoparticle carrying mRNA gets into the cytoplasm of cells in this mice, and that mRNA turns into protein, that protein then translocates into the nucleus and can kind of excise that lock-stop-locks cassette so that we get TD tomato expression in those cells. And so what this allows us to do is specifically sort for TD tomato positive cells, and thus sort for LNPs that deliver their mRNA payload successfully to a cell. So this technology was really influential and kind of impactful in the direction that the research that we did in the Dahlman lab at Georgia Tech went, and it allowed us to find lipids that deliver to splenic endothelial cells, as well as novel phospholipid structures and novel cholesterol structures. And it also kind of opened the door for potential delivery to lung and heart endothelial cells, and the potential to identify novel lipomer structures and novel formulation ratios in phospholipids. And so while this system was really, really great, it did have a caveat, which is that you're stuck with screening in these AI-14 or AI-9 mice. And it's not easy to translate this system. In fact, it's impossible to translate this system as of now to a non-human primate animal model. And so what we did is we created SANS, which is a species agnostic screening method. And this was done by Marina Kadit and other colleagues in the Dahlman lab. And basically the way that this system works is that particles are still formulated with an individual single-stranded DNA barcode. But in this case, instead of using mRNA, we use something known as AVHH mRNA. And AVHH mRNA codes for AVHH protein, which isn't regularly found in mice, non-human primates, rats, or humans. And so this the expression of this protein can easily be kind of identified using common flow cytometry techniques, or as we'll get to down the line, some other techniques as well. And so, for this system, what we did is we injected pools of LNPs into normal black 6 mice, as well as humanized mice, which were basically mice that had repopulated humanized livers. And we also injected this into monkeyized mice. So these are monkey cell repopulated livers in mice. And what we found was that there were differences in terms of transcriptional response to lipid nanoparticles depending on which mice you used. And so if you're interested, go ahead and give this paper a read. It was published in Nature Nano last year. And so these systems are really great, but I think, they don't address some of the issues that I kind of brought up at the beginning, which is that if you're trying to hit rare cell types, it can be really, really difficult to do that using existing methods. And so what we did is we created a new screening system, which is called SENT-Seq, or single cell nanoparticle targeting seq. And SENT-Seq is basically a high throughput LNP screening approach that combines the additional informational toolbox that's given to you through single cell RNA sequencing. And so the goal of this whole project was to basically identify LNPs that deliver mRNA to rare and hard to classify cell types. So this can be HSCs, it can be lung basal cells, basically any cell type that has that there's a lot of controversy as to how to particularly characterize these cells, at least on the flow side, but maybe less controversy when you can look at their transcriptional markers. And so we wanted to do this in order to try to find novel LNPs that could potentially treat disease. And so in this particular approach, we utilize single cell RNA sequencing in combination with DNA barcoding. And this approach allows us to characterize rare cell types based on their transcriptomic profiles instead of their flow profiles. It also allows us to identify potentially immunogenic and toxic LNPs that deliver mRNA. And I'll kind of get into at the very end of my talk how we're starting to do that. And so the way that this approach works is very similar to all of our other approaches, at least at the beginning. So particles are formulated so that they contain their own individual single-stranded DNA barcode and that same AVHH mRNA that was part of the SANS system. These particles are then pooled and administered into mice. And for the purposes of this particular paper, we stuck to the liver because we know that we have a lot of good delivery vehicles that go there, and it was a good proof of concept. So we had to do a lot of improvisation on the molecular biology side of things and the computational side of things. So it felt like the best proof of concept to us. And so we adopted a single cell RNA sequencing method, which was recently published in Cell, known as microwell. And since then, BD has created a system that kind of takes this and puts it into a kit, and that system is called Rhapsody. But we used microwell for the purposes of this paper. And the way that that works is that you have a device, and imagine that device has 1 million different wells on it. And so you load your single cell suspension. In this case, we had a single cell suspension of liver. We didn't enrich for any cell populations. This is just pure liver, basically digested and brought down to a single cell suspension. And we load the cells onto this microwell device at a concentration such that there's about 1 cell per well. And the reason for this is that you want to try to avoid doublets when you're doing single cell. Once we load the cells, we load these special magnetic beads onto the device such that you get about 1 bead per well. And so each well has a cell and a bead. And these beads are special because they have a poly-A binding site and they have an LNP barcode capture site. So once you've put your beads and your cells on your device, you then lyse these cells, and all of the mRNA, all of the barcode that is in each of those cells kind of spills out and can bind to the particular adapter on these beads. We then do single cell library prep, and we can read out which cells we have in our population using a t-SNE or a UMAP. And on top of that t-SNE or UMAP, we can overlay delivery for all N of the LNPs that we're testing and all of the cell subsets that we're using. And then on top of that, we included an additional component to this, which is that which basically allows us to also read out how much protein expression of our AVHH protein we have in each of these cells. And so not only are we getting LNP barcode delivery biodistribution, we're also getting the amount of protein that's being created in each of the cells where we also see those levels of barcode. And so we have biodistribution and functional delivery all at the single cell level. And so what this looks like in practicality is for this particular experiment, we identified, I think this is about 15 or 16 different hepatic cell populations. And so you can see all of those here, and they're color coded. And what you see is that we're picking up different types of Kupffer cells, and the way that they're differentiated is just based on their gene expression profiles. We're picking up multiple types of hepatocytes, endothelial cells, cholangiocytes, and some other types of cells as well. And so what you can then do is you can overlay AVHH protein expression onto each one of these single cells in our particular experiment. And so again, what this allows you to see is the amount of functional delivery that you're getting in each cell, because AVHH protein is the result of the mRNA that we're delivering inside of our LNPs. And then on top of this, what we're really excited about is that you can overlay the expression and biodistribution of each one of these different LNPs in our screen. In this particular case, we had 24, and you can start to pick up trends about where these LNPs are going. So for instance, you see that this large cholangiocyte cluster has a lot of LNP-10, and then down here, you can see that some of these Kupffer cell clusters have a lot of LNP-1 and LNP-7. And so we analyzed this data, and we looked at the 4 most abundant LNPs in our screen in terms of their biodistribution and functional delivery profiles. And so we picked up, these 4 LNPs. And again, the structures of these LNPs are not anything special. A lot of these components have been out there for a while. And so we weren't surprised that these particles worked well. And what you see is that, across the board, you have pretty good LNP biodistribution with most of these particles. So that's the first thing. The second thing is that a lot of this biodistribution is not cell type specific. So I think that's something that's important in terms of lipid nanoparticle delivery in general, is understanding tissue specificity. So how often are you getting to your on-target tissue, like the liver or the lung, and how often is your particle going to your off-target tissue? And then even within that tissue, does it matter that you're not getting specificity for a particular cell type? So if I only want to hit endothelial cells, none of these particles would be a good candidate, because they sort of seem to at least have biodistribution everywhere. The other thing that we noticed is that LNP-10 seemed to kind of cluster in cholangiocytes, but had lower barcode expression across the board, whereas LNP-12 seemed to go to less cells in general, but have really, really high barcode expression in the cells where it was found. And so on top of this, we also overlaid AVHH protein expression. And so what you see here is that for LNP-3 and 7, the protein expression kind of mimics the biodistribution, in the sense that where the particle's barcode was found, we're also picking up a lot of a lot of protein expression. LNP-10 is of note here because the protein expression is actually quite low compared to, some of these other cholangiocyte delivering LNPs like LNP-7 and LNP-3, even though the barcode biodistribution wasn't quite that low. And finally, we can see that with LNP-12, we have high biodistribution and pretty high functional delivery in those particular cells as well. And so we really were interested in kind of seeing exactly what was happening with LNP10 and why that protein expression was so low compared to some of the other LNPs. And so this is where our aim at kind of looking at immunogenicity and toxicity came into place. So we took a list of about 300 inflammatory and toxicity genes, and we looked at their RNA expression in the cells where we were getting delivery of LNP10. And what we found was that there was upregulation of a lot of inflammatory genes, as well as upregulation of at least one toxicity gene, caspase-3, in the cells that were getting this particular LNP. And so, of course, there's, a lot more work that needs to be done to kind of use this as a screening method for toxicity or inflammation. But we think it's a pretty good proof of concept, and we're really excited to kind of develop more data and look into the prediction capabilities of our SENT-Seq platform in the context of inflammation and toxicity. And so finally, our SENT-Seq platform allows us to screen, in this particular paper, 24 particles at once, but we've done over 150 at once in other tissues. It also allows us to discover new LNPs that target hard-to-characterize cells, because we can characterize cells based on their transcriptomic profiles instead of having to use flow cytometry antibody-based approaches. And using the transcriptome, we can determine if LNPs are potentially toxic or immunogenic during early development as opposed to during later development. And this technology is, as I mentioned, being licensed into our new startup, Nava Therapeutics. Finally, I'd like to thank everyone who was part of this work, as well as all of the work that I showed. All of this work was done at the Dolman Lab with a lot of these folks. And I'd like to thank our collaborators in Philip Santangelo's lab and our funding sources. And I will open the door for questions.